What a sheer joy to have the privilege of introducing Dr. Stephen Ansell, world-renowned WM expert at the Mayo Clinic in Rochester. As you guys know, Dr. Ansell's chair of the IWMF Scientific Advisory Committee. He's had that role since 2015. He is also on the editorial board of the Journal of Clinical Oncology and associate editor of American Journal of Hematology. He's a member of the scientific committee of the Leukemia and Lymphoma Society, which we're all well aware of, and chair of the Mayo Clinic Lymphoma Group. His research is focused on understanding the biology of B-cell malignancies and exploring the role of the tumor microenvironment in supporting cancer cell growth and survival, with the goal of translating this knowledge into new treatment approaches for patients. So, if y'all have had the honor of hearing Dr. Ansell present at an ed forum in the past, you know that he is an all-time fan favorite presenter and a master educator in WM. Dr. Ansell has a unique gift of making the complex and the confusing seem straightforward and comprehensible. So, true story, I need all science explained to me like I am a fourth grader. And if you've seen Dr. Ansell and his slide deck with his fun images, relatable metaphors, and understandable language, you know that everyone always walks away with 100% clarity, which is no small feat in the world of WM. It's such a gift. So here's what we're going to cover today during our time together. Dr. Ansell is going to explain the critical work of the IWMF Scientific Advisory Committee, which our support group leaders have never had the opportunity to hear from a committee member. So this is a huge honor, lots of excitement. Number two, he has agreed to discuss how the COVID vaccine might impact WM patients. Not a hot topic at all, right, guys? Oh my God. Number three, he is willing to answer a host of fascinating questions that were submitted by all of you in advance. Thank you for your questions. They are very high level, highly informed, and really impressive. So it'll be a fun conversation. And lastly, we're going to wrap up today with asking Dr. Ansell to answer our critical question that we're asking of all WM rock stars, which is, what is the one thing that you wish every WMer knew today? We like to spring that on you without prep to see what's top of mind, but we like you so much that we didn't want no surprises for the doctor. So we decided to give you the cheat sheet in advance. And Dr. Ansell, the floor is all yours. You have our enormous gratitude. Thank you so much for everything that you do 1,000,000 times over. We WM patients and everyone around the world is so grateful for both your research and all the work of your hands and your heart. It's all yours.
Well, thank you very much. And again, really, thanks for everyone being on. My goal today is to make this fun, hopefully informative. It's kind of a high bar you set there about everyone being completely clear, because I'm not even sure I'm always entirely clear, but I'll, fake it till you make it, right? So I'll just keep on. My mom always used to say, when in doubt, sound convincing. So I'll do my best on that one. But what I'm hoping is that you folks will find this valuable. Thank you for your questions. And then Lisa said right at the end, we'll probably have some time for open mic, and I'm totally open to questions that you might have, because I think, again, you are advocates for patients, for colleagues, family members, and you are really kind of the way in which the IWMF reaches the world to share with them what they need, what people need to know about Waldenstrom. So thank you for what you do. And I'm hoping that the little bit of conversation we have today helps you just have a more informed view of the work that you can do and are doing. So again, thank you so much for what you do. So with permission, I'm going to share my screen here, because I've got a few slides I want to show, just because, again, I'm a person who likes to learn by listening and seeing, and my attention span is usually short. So first question I have is, can you see that? So Lisa, I need you to tell me yes or no. I'm hearing perfect, but all's muted.
Yes,
okay.
We can see it, Dr. Ansell. Thank you.
Perfect. So as you said, what am I going to talk about today? So I'm really going to cover 3 topics, and then there's a bunch of questions you gave me, and I'm going to happily go down that list and answer them as best possible. And then at the end of the day, I'm going to, with Lisa's permission, just kind of have an open mic. If things have come up from what we discussed that you want to address, happy to do that. So talk about what the Scientific Advisory Committee, otherwise known as the SAC, what is it and what does it do? And why do you need it? And then talk about COVID and the COVID vaccine. And what I want to do there is really share with you what exactly the COVID vaccine is doing to your immune system and how it might impact patients. And there are a number of questions we'll get to afterwards as well. And then the question that everyone's being asked, what's the one thing that I wish everybody who had WM or knows somebody that has WM knew today? And hopefully, as I say, that will be helpful to you as well. So what's the SAC and what does it do? You know, SAC, unfortunately, is a kind of an unfortunate acronym. I came from a different part of the world where acronyms are not really a thing. So I spent all my life kind of shaking my head going, well, what is that exactly? But really, Scientific Advisory Committee, that's what it is. So really, it's not the picture on the left, it's the picture on the right. It's a group of people that are going to get around and talk about what do we need to be aware of and what does the IWMF need to be aware of when it comes to Waldenstrom's. So it's really a bunch of experts. So again, compliments to Newton and Carl and others before who really engaged many experts from around the world to be part of that team. Dr. Kyle also was key to that right in the beginning, to bring these folks together, and they do a number of things. So the first is, I think they provide scientific insights, oversight, and advice and direction to the IWMF. And I'm going to touch a little bit on the roadmap that many of you are aware of. But again, I just wanted to remind you what that's about, why it matters, what pieces of information it provides, so that as you talk to your friends and colleagues and other patients, you know that. So they kind of give the advice, number one, but then they also identify the knowledge gaps. And I'm going to focus on that a lot because the knowledge gaps are really what this roadmap is about. Because again, if you aim at nothing, you hit it every time. The goal is if you don't know where you're going, you don't know if you're getting where you want to go. And that's really what the knowledge gaps are about. Many of you are very instrumental in supporting IWMF and particularly research and funding for research. And so the SAC plays a role in overseeing the grant process, reviewing the grants, and along with the research committee that some of you may be part of, it really kind of helps with the whole grant sort of process. And then these are people that are interested in Waldenstrom's. These are people that actually know about Waldenstrom's, and they are advocates for patients, but also for the disease all around, and in their areas of influence bodies to pay attention to Waldenstrom's. And so again, I think that's in essence what the SAC overall does. So the key thing is, I mentioned knowledge gaps, and I think that really has been, and I want to compliment the IWMF, that's really been an area where the IWMF has played a major role. Right back in the beginning, we kind of knew nothing about Waldenstrom's. We really did in Waldenstrom's whatever we thought was good for either myeloma or CLL or lymphoma, and surely that would be good for Waldenstrom's. And actually, as we've identified these knowledge gaps and worked on focusing on them, we've learned that Waldenstrom's has a lot of very unique features, unique biology, and therefore unique and useful treatments that have made it a much more specific entity and a much more effectively treated entity. So actually, this has been a major contribution the IWMF has made to the world of Waldenstrom's, identifying the knowledge gaps, supporting the research, getting some of the answers. Note I said some. We don't have all of them yet. So that's really kind of the key part why this is still an ongoing endeavor. So you've heard this before. Going to touch on these things again. What are some of those areas where knowledge gaps exist? And really the roadmap group has spent time thinking about this, and these are the key areas that are felt to be important. We really need to understand more about the signaling, and that's how the cell talks to itself and how it talks to other cells around it. And I'll share a little bit more about that in a minute. Furthermore, it's about what we call omics, and that's because you get all these fancy words: genomics, epigenomics, transcriptomics, proteomics, metabolomics. And what that means is the way in which the cell processes information. And again, this is important because we've understood quite a lot about the genomics, the code, but there's a lot downstream of it that we don't. And I'll talk about that in a second too. The other thing that's important is the immune system is kind of like just watching. It's not actually getting out there and helping fight the cancer like it should. And so it's a little bit like, you know, cops on the take who are just turning a blind eye while the shoplifters are going at it. And again, how can we make the immune system wake up and do a better job of picking out the bad boys? And then finally, sometimes the environment in which cells exist is because they like it there. And it's really interesting to me. I learned this right in the very beginning. If you take a Waldenstrom cell out of the bone marrow and you try and grow it in a dish, it doesn't grow. In fact, I can tell you for a fact, we did 100 cell line development projects to get one cell line. So it is very difficult to get a cancer cell to grow outside of the body if you have Waldenstrom's. It grows inside the body just fine. So the question is, why is that? What about the environment could we know more about so that we could change it? And really, I joke and say it's like having a crack house in your neighborhood. Your neighborhood is never going to be better until the crack house is gone. So you better go and do something about moving those people out. So signaling is the first thing. And again, I'm showing you a picture here of what goes on inside a cancer cell. And these are all, every one of them, key pathways within the cancer cell and the way in which the cell is talking to the environment around it, kind of a messaging system that goes down into the nucleus to generate all kinds of proteins and things that it needs to do. And you might look at this and go, holy smoke, that is just crazy, out of control. How on earth do you make any kind of sense of that? That is just nuts. But I would actually put it to you that you do that every day. If you live in any kind of big city, you have, and this is the London Underground, you have, you know, a map that looks just like that. And you know which path you need to get on and which station you get on and where you change and where you get off. That's all pretty straightforward. And you've worked it out. And so again, as we understand this better and better, we can actually work out where exactly we can change things, because that would then disrupt processes that are detrimental to the patient and promote ones that would actually make things better. So the whole thing of signaling, as we understand this map much better, that actually is a way in which we can really fundamentally change how we do things for patients and make things better for them overall. Then the whole thing about omics. So again, this lady saying to the guy, do these genes make me look fat? And to be honest, the answer is yes. And again, the reason you're fat often is because of your genes. And so no judgment for anybody, but I think what's very important is a lot of what happens in your body is driven by genetic code. And as you well know, in the world of Waldenstrom's, really defining the disease in many respects is a mutation, a change in the code in the MYD88 gene. And because of that, that then causes those cells to basically have a lot of messages that are amplified. And because they're amplified, they make the body and the cell behave in a way it shouldn't. And that's what drives Waldenstrom's. But there's much more to the story than simply that. So there's a whole thing then about the genomics and then the epigenomics and the transcriptomics and the proteomics and the metabolomics. And you would go, again, that's nuts. Way too much information. I don't understand this. But again, as I'm going to illustrate, you do this all the time, every day. So for example, on the left, you can see, maybe you can see my arrow here. Here is the gene code. That's the genomics. But again, the way in which your body works with the gene is it makes copies of some of the genes, not all of them, just some, so that we can use that information. It's again, you don't take the entire textbook and read it cover to cover. It's like an encyclopedia. You turn to the page about the topic you're interested in. So the same thing is happening. Your body goes to the genes it cares about right now, the cell does, and it does what's called transcription. It makes a copy of just that gene. And that part of all the copies it's making, that's a subset of the genes, and that's called the transcriptome. And then what it does is that code of only the genes it cares about, it turns those then into proteins. Because again, the genes don't do the work, the proteins do the work. But as proteins then get moved, as you well know, proteins are metabolized and they are turned into metabolites. And the metabolites are what do the work. So here's your genomics, transcriptomics or epigenomics, proteomics, metabolomics. And you might go, wow, how much do we know? Well, we know quite a lot up here, but we kind of get a lot more vague as we go down here. And this is where there's a significant knowledge gap. Now, again, you might go, this is making no sense to me. Well, no, it is, because this is how it works in your life. You have a computer. If you have nothing on the computer, it's a very expensive doorstop because it just stands there. It does absolutely nothing. You need to load programs onto it to actually be able to do something. So let's say, and it just got discontinued, so maybe this is a silly use of this, but Flash is one of the ways in which you use a program on your computer. And when you do that, often you would go to something like YouTube, and then on YouTube, you would watch your favorite cat video or whatever. So again, the point is just, it's going from the equivalent of genomics, transcriptomics, proteomics, metabolomics, to take it from code that you can't understand to a picture of some cat dancing on the piano that everyone thinks is hysterically funny. So you heard, firstly was the whole story of signaling. Secondly was the whole story of omics. The third story is about immunology. So remember I said your immune system is taking a nap when it really shouldn't, and it should be taking on the cancer cells in a much more effective fashion. So I'm showing you here some little fun pictures of all different kinds of cells that are present within your immune system. And your immune system is actually quite complex with a lot of different cells that tell it whether to go after something or back off from something. So if you can imagine, if you get a pimple on your arm, you know, you initially want, when there's some bacteria in there, for your immune system to come rushing in and to grab a hold of those bacteria and beat them up and kill them and wipe them out. But then you want it to stop, because if it keeps on fighting and does it, you're going to end up with a crater in your arm, because that's all going to get broken down. So you actually want things to back off and then healing to happen and a little, you know, sort of, if needs be, a scar to develop and everything just to kind of close over. So you have all these different cells. I won't go through what each one of them do, but I think there are a few ones that are interesting. So you see up here on the left, a cell called an NK cell. So an NK cell is a natural killer cell. I joke and tell people, this is kind of like the bouncer at the club. This is the guy who stands at the door, and if you look like you're misbehaving, he's going to just take you outside and slap you a few times. So not very sophisticated, somewhat grumpy, just big and burly. And so in essence, NK cells are cells that can be very quickly mobilized, but aren't particularly highly specialized. There are what they call cytotoxic T cells. These are green berets. These are really well-trained, you know, folks that can go after and slit throats in the middle of the night, you know, with no one even knowing they were there. Very specific, very effective. The helper cells are kind of the logistic guys. You don't get anywhere, you don't get the supply chain to your frontline troops unless you got T helper cells. Follicular dendritic cells are kind of like intelligence. They're the ones who gain information and then share information with the immune system. Macrophages are trash collectors. They basically eat anything. If you ever watched like Ratatouille or one of those sort of movies where, you know, the rats who were eating kind of junk, weren't even sure what they were, those are macrophages. They will pretty much eat anything. And then when they sort of halfway through chewing it, they'll go, no, I wonder what that was. But they'll present it to the immune system so the immune system can know whether that's something that's a threat or not. Treg cells are cells that control other cells. So again, if you don't want a cell to get out of control, this is who kind of maintains the peace, if you like. And then I'm just going to take 2 seconds and talk about these two guys, because this is what matters to folks with Waldenstrom's. If you were to breathe in a virus, a lot of the cells, like the NK cell, would grab it, the macrophage would eat it, the dendritic cell would present it. Sorry, and this B cell then would be one that would be developed specifically to target the virus. In other words, it would begin to make antibodies. But what it does is it starts off making antibodies and it changes from a B lymphocyte to a plasma cell. And the plasma cell is the one that you can see there from the picture is spewing out lots of antibody. As that happens, you move from an IgG, an IgM to an IgG molecule. So when people say I have Waldenstrom's and they mention they have a lymphoplasmacytic infiltrate in the bone marrow or a lymphoplasmacytic lymphoma, you can hear the word lymphocyte and plasma cell. It's the process going from lymphocyte to plasma cell where the cancer cell got stuck. And that's exactly where IgM is made. So you haven't switched to IgG, you're stuck at IgM. And the reason that matters is IgM molecules are like the NK cell, the first response, quick and dirty. It's a big molecule, it's pretty sticky, it'll stick to a lot of things, but it's not very refined yet. It'll get much more refined when the plasma cell takes over. But remember, then that's what causes the symptoms. It sticks to lots of different parts of your body, causes neuropathy, causes anemia, and then the blood gets thick, your blood then causes hyperviscosity. So again, that's where a lot of it makes sense, a lymphoplasmacytic cell making an IgM protein. So I think, again, I mentioned this before, but we want to now really get the immune system, and this is what we understand better. Why is the immune system not seeing the cancer? And if we then could make it do better. So in essence, you can see that dendritic cell, the follicular dendritic cell, is kind of going to the cytotoxic T cell and the helper cell to say, I'm like the scout, I saw some trouble. I'm just coming to share with you what I saw was this weird little virus. So you might want to look for that. And then these cells will actually then talk to each other, send out a party to go and hunt down the virus and take it out. We want to obviously do the same thing where it will hunt down the Waldenstrom cell and take it out. I'm showing you across the bottom the whole story of CAR T cells. We often get quite a lot of questions related to that. So if you take that cytotoxic T cell and those T helper cells, and you actually put a viral vector into them. So in other words, what that is, it's a small piece of gene code, goes into the cell incorporates it into its genetic makeup, and it starts making what that code is coding for. And that's usually a docking site. So you can see right here on this picture, this green thing on its head, that's a binding site or a chimeric antigen receptor. And you can see as those sort of stick out, it makes this a muscle cell, if you like, a bigger, more aggressive cell that's able to detect the cancer more efficiently and actually target and kill the cancer. And, there have been a number of studies now looking specifically at low-grade lymphomas and showing that that's an effective process. Lastly, as Aesop from Aesop's Fable said, a man is known by the company he keeps. So, cancer cells tend to hang out in the environment in the bone marrow. And somehow that bone marrow has got some guys that are facilitating bad behavior. And the cancer cell is behaving badly. So if we can change the environment, clean it up, as I mentioned earlier, get rid of the crack house, that will actually make a big difference to who wants to live in the neighborhood, and will get bad elements like Waldenstrom cells out of there. So what I'm hoping you're hearing us say, or hearing me say, is multiple reasons the SAC matters. Scientific Advisory Committee advocates for Waldenstrom's, ways in which they then would give guidance and oversight to the IWMF, help with the grant reviewing process, but set the direction for Waldenstrom's research, identifying these key gaps in knowledge, and then sharing with the community and hoping for funding to be put to actually answer those questions. And again, compliments to the IWMF because great progress is made, especially in the omics area, and now more so in the environment, microenvironment side of things, and more in the immunotherapy. But there's still a lot more work that needs to happen in that space. So the next thing I wanted to just take a few minutes to talk a little bit about COVID vaccine and how it might impact Waldenstrom's patients. We get these questions a lot, and it's not just Waldenstrom's, it's any kind of cancer patient. In fact, our cancer center put out a statement just a little bit ago which basically said their recommendation, and I'll share a little bit more about this, is that every single patient with cancer get the COVID-19 vaccine as soon as it's available to them. And we'll talk a little bit more about that in a minute. So here's the COVID-19 virus. So just again, to make sure everybody understands what a virus is. So a virus is a very simple particle. It doesn't even have a brain. So when people think the virus is picking on them, well, it doesn't have a brain to even pick on you. And when they go, well, you know, it's picking on me for whatever reasons, political reasons, not whatever, not true. It's entirely agnostic. It doesn't care, and it doesn't have a brain to think. It's just a piece of code. So in essence, what it is, it has 3 bits to it. It has this little spike protein, this little red thing that you see, and that just helps it stick to cells. Then it has an envelope. That's the kind of round ball part. That's just to protect the actual code inside from the various digestive enzymes in the blood that would break it down. And inside it, it just has some code. That's all it is. So what this does is it sticks to the cell, it goes into the cell, it copies the code, and it makes the cell, it hijacks the cell to make lots of the same code. So I show this here on this picture. You can see here at the top, the spike protein. Here's this kind of capsule or the bit around it. There you can see the code inside. So what happens is it sticks to what's called the ACE2 receptor. That's the docking site on the outside of a cell. It basically gets sucked in. It then takes its code, goes into the nucleus. The body just makes a whole bunch of copy. It kind of just gets part of its natural process. It just starts making lots of copies of the virus. The virus then just gets packaged by the cell and then gets sent back out again. So pretty much all the virus has done is it's just taken over kind of how the cell works. It just inserts into its code exactly all the code to make its own virus. So that's why computer viruses are called viruses. When you have a computer virus infect your computer, it gets into the hard drive and it just starts to make you send out silly messages or emails that aren't from you, or basically crashes your computer. And in essence, that's what this virus is doing. Gets into the cell, utilizes the same processes, just makes thousands and millions of copies of itself. Those all get packaged up and released, and then the cell dies. And when it does, millions more of these viruses go pouring out. So how does the vaccine work? So for years, the way we used to do vaccines is that we took the whole virus and what we call crippled it. We basically just stopped it from working and injected that in. And then the body went, oh, that's not good. And then immediately made antibodies against something that was foreign to it. But the current viruses current vaccines that we're going to talk about actually are even cleverer than that. And that's part of the reason why this whole warp speed thing was really warp speed. So basically all the vaccine does is it actually does exactly what the virus is doing, but just not in a way that actually can really make you sick. So remember, the vaccine looks pretty much the same as the virus. Again, it also got a coating around the outside, and it's also got a code on the inside. Interestingly, this code doesn't code for the whole virus. It just codes for that little spike protein, the little bit that sticks on, those little red bumps that I showed you on that picture. But it does the same thing. They inject it into your muscle. That's why it hurts. It gets sucked into the cell. The coating goes off. The code gets translated in exactly the same way. But now instead of making viruses, you just make those little red bumps. That's it. Just the sticky bit. So when you make that little sticky bit, the body goes, that's not good. But it's really reacting to only part of the virus, not the whole virus, because you don't have the whole virus. But you'll make the same immunity because that particular thing is very worrisome to the immune system. So that's also why you're hearing a lot about these variants, and the worry about that is because what's happening in these variants is that this little code is changed a little bit. And so the one that comes from England and the one that comes from South Africa, where I'm from, so sorry that somebody from South Africa is sending us something we don't want, but they have mutations or changes in this code, which makes us a little worried about whether this protein will still cause the same immune reaction, yes or no. So the last thing I wanted to, and we'll talk more about, there are lots of questions about vaccines, and I'll cover them and then happily take your questions. What's the one thing that I wish every WMer knew today? And I would say I started doing, I qualified in 1986. I started doing cancer therapy and cancer treatment in about 1990. And when I started, and you asked me, how long is an average patient with Waldenstrom's likely to be alive? The answer was like, 3 to 5 years. So I wanted to show you the survival curve of patients with Waldenstrom's. And this is now not people that have MGUS. This is people who are sick and need to get treated. How do they do today compared to how do they do back many years ago? And you can see here, this is the outcome of patients who had, this is now not 10 years, 15 years ago, but you could see that that line was heading straight down. In about 5 years, half the patients had had real trouble. But if you look at the data now, that actually is substantially different. We actually haven't hit the 50% mark yet, and you can see we're out now, on average, we think well beyond 10 years. So again, I'm an optimist in life, and I tell people we never want to be average. You always want to be well above average. So if you're above average, you aren't even on this curve yet. So bottom line is, new therapies, new ways of treating and thinking about the disease are making a substantial impact, not just on how people do, but how long people live. And my hope and prayer is that as we continue to invest in treating patients with Waldenstrom's, this line will eventually just be one that's right up at the top here. In other words, decades go by and people aren't dying because their treatment either cures the cancer or completely controls it, meaning it's, like having blood pressure or something. So again, I think for me, this has been a massive change where when I see patients, rather than having to give them, kind of a concerning news, I can say there are lots of tools that can be very effective in controlling the disease. However, I think Newton, Carl, Pete, others have said the goal is to get to a cure. So we can't look people in the eye at this point and say, we can cure you for sure. But we can look them in the eye and say, we can treat you so effectively that the outcome of patients is dramatically better. So I think for me, that's the one thing I wish every WMer knew today, is that the outcome of what WM is profoundly better than it used to be and improving all the time. And it's largely due to the contribution of folks like you. So that's why I just wanted to say thank you for what you do to support IWMF. And there are a bunch of questions that I'm happy to answer, but maybe what I'll do is turn it back to Lisa if you got anything right now.
Thank you so much, Dr. Ansell. That was amazing. I'm so grateful to you. Yes, bravo. Everybody's already clapping. Truly. Thank you so much. Eye-opening, enlightening, and you're right. I understood it even better. So I'm going to adjust the introduction and not say 100% because that doesn't allow any room for improvement. And I understood it better this time. So I didn't walk away with 100% last time, clearly. But today I did. So thank you for that. And why don't we see if there's anybody who might have some questions specifically about what we just did. But remember, folks, you submitted so many great questions in advance. I also want to be sure we get plenty of time for those. But if somebody had something burning that they really needed to have addressed before we went on to our pre-submitted questions, what do you think, Dr. Ansell? Should we just take a minute?
I'm open to anything.
Oh, brilliant. Thank you for your flexibility and openness. What do you think, guys? Just taking the temperature here. Anybody super confused about something or want to see another one of those cute Sal cartoons, because they're personally my favorite because it allows me to understand it. Fourth grader. Pat.
I think go to the pre-addressed questions and then see if anybody has questions.
Okay, I'm seeing agreement. I'm seeing nodding heads. Pat, Great for jumping in there. Let's go right to the questions that we have submitted. And at the very end, we'll catch up with any that are still burning. Good idea. Dr. Ansell, what do you think? You want to take it away in whatever order, whichever ones speak to you there?
I'll just go straight down the list so that everyone's questions that were asked get answered.
Wow.
And again, I'm not always sure I have all the answers, but I'm certainly willing to give it my best crack. So the first question I was asked was, so there are a bunch of COVID questions. So I think we'll start right off with those.
Great.
Can you get reinfected and transmit COVID-19 after you're recovered from COVID? And the answer is, we're learning a lot about this, and we know that in part the answer is yes. So there are people that have actually been reinfected. There are people who got vaccinated and got infected. So it's not a guarantee. However, it certainly decreases your likelihood of getting infected dramatically. And particularly if lots of people are vaccinated, that actually really will help overall. You hear this talk about herd immunity. If the virus isn't being passed around a lot, then it actually peters out, because you notice it doesn't live on its own. It's just a piece of code. So if it's got nowhere to go and no one to be shared with who can receive it, it's going to go away. So quite frankly, if we wanted to get rid of COVID-19, if we could all just never talk to anybody else and just stay in our houses for like a month, nobody in the world move for a month, it would be gone, because that's how long it takes to burn out. The problem is people just can't stay in their houses. It's just not physically possible. But quite frankly, that's why you heard the whole lockdown in the beginning. They were trying to do that, is just don't go anywhere, because in 2 weeks, if it doesn't get transmitted, it goes away. So if you just don't go anywhere, it wouldn't be transmitted. The challenge, unfortunately, is, people rob banks in that time, so someone's got to go stop them. So that's where it ends up being much more complicated. So then the next question is there added risk of the COVID-19 vaccine for highly allergic or sensitive people who have reacted to Rituxan or IVIG, etc.? And the answer is, don't know. What we do know is this is a different reason to be sensitive. So if you were sensitive to IVIG or you've been sensitive to rituximab, that doesn't mean you're going to be sensitive to the vaccine, because the way in which it's made and what you are reacting to is different in those two circumstances. So some people are very reactive to lots of things. So yes, it could happen, but the actual mechanism of why you're reacting is different. So if you had a reaction to Rituxan, doesn't automatically mean that you're going to react to getting the vaccine. In fact, most of the time, the reactions to Rituxan have to do with the pace at which it's given to you. The slower you give it, the less you have a reaction. So that's actually not a real allergic reaction. It's a hypersensitivity reaction. And it's really because of the foreign protein that you're receiving. This is not protein, actually. This is actually an mRNA, which is a different part. This is the code, not what the code makes. What's your opinion on receiving a COVID vaccine, whether before, during, or post chemotherapy? I tell patients the time to get your vaccine is the moment you can get it. So it's not a case of when did I get chemo last, where am I? If you can get it, get it. And if you can get it, tell your family members they should get it. Because quite frankly, if they are immune, they're not going to pass it on to you nearly as easily. So I would say this, that if you just recently got chemotherapy, if you recently got rituximab, your take rate, the likelihood that it's going to work is not 95% like is in the study. You're probably lower than that, but it's not zero. So that's the reason to get it, because it's going to help to some degree, and it's better than nothing. And quite frankly, a lot of people say, well, what about the side effects of the vaccine? Well, those are generally way, 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 way less than the side effects of getting the infection. So I think in my mind, it makes no sense if you have it offered to you to decline. Should one get the first shot or the second shot within days of treatment? What about people on ibrutinib? I tell people, just get it. Get it whenever you have it available to you. Doesn't matter what you're on. Doesn't matter whether you got it or you didn't, treatment-wise, just get it. Could I wait and see if the first dose, or if the 1-dose COVID vaccines work better than those that are already approved? Because again, most of you will know everything that right now you're going to get 2 doses. So personally, I just got my first dose about 10 days ago. I have to wait another 10 days to get the second dose. The immunity is about 70% after the first dose, and it goes up to about 95% after the second dose. So all told, I don't know right now when the 1-dose vaccine is going to be available is somewhat delayed. For me, it doesn't make a lot of sense to wait for something when you are just putting yourself at more risk of getting COVID-19 in the meantime. You know, most parts of the country right now, COVID-19 is out of control. So you are at risk of getting it. And if you have the vaccine offered to you, my recommendation would be get it. What about the AstraZeneca one? So that's a virus that's a weakened adenovirus. Is that safe for Waldenstrom's patients? Not been tested, but theoretically, there could be that that virus would actually maybe cause some problems in you. But I will tell you that they entirely have crippled that virus. So that virus is not actually something that can replicate very well. And so I would say whichever vaccine is offered, I would just go ahead and take it. The only thing that I would say that there's some information, this comes to one of the questions, what about, you know, other vaccines? If I'm offered the shingles shot or the influenza shot versus the COVID-19 shot, we tell people 2 weeks in between them. So if you're getting the shingles shot, don't allow a 2-week break before you get the COVID-19 or vice versa. So I think just looking down the list, if you've had a stem cell transplant and you're taking drugs like valcyclovir and other things, will that be a contraindication to the vaccine? No, because again, remember the vaccine is a code, not actually a virus or anything at this point. I think those are all the COVID-19 questions, and the rest of the questions we have here are about Waldenstrom. So maybe, Lisa, I'll turn it back to you in case we want to just ask some more COVID questions, and then we can go to the Waldenstrom's questions.
Thank you so much, Doctor. We have one in the chat box from Betty McPhee in Canada who's asking, does a COVID-19 test only test the COVID virus, or does it also test the flu and or cold?
Yep, so when you basically have a test, it's called a PCR, polymerase chain reaction test. It's testing very, very specifically for that RNA sequence. So when you have a COVID-19 test, it only tells you whether you had COVID-19, if you have COVID-19, yes or no. It tells you absolutely nothing about anything else. And to be frank, it's not 100%. So if you just got infected today and you're not making too many copies of the virus yet, it could read negative. So that's why, again, just being aware that it's a useful test. It helps you have some sense of it, but it's not a perfect test. And it certainly doesn't tell you anything about other diseases. And so remember, a lot of people talk about flu, and they mean everything from like having upset stomach, diarrhea to a runny nose. Most of the time, those are adenoviruses and Coxsackie viruses. They're other things. Influenza, if you had that, you remember it. You felt completely washed out, had a really high fever. You felt sick for a week. And so COVID-19, as you know, if you get sick from that, not everybody does. Some people have really a minor illness, but some people have been deathly ill. So again, it's the same thing. These viruses are kind of a little sneaky. So your PCR test is very useful, but the only thing it'll tell you about is what you're testing for.
Thank you, Doctor. I know Mel Horowitz in New York has a question about COVID. And then Shelley, I'm just planting the seed with you. Were there, after Mel asks his, if there was anybody else who sent us late questions submitted about COVID, I'm forgetting if there might be anyone. I just thought I'd mention that.
I know someone had asked about the shingles, but he just addressed that. But let me check after Mel's question.
Mel, let's see, are we able to get you unmuted? There you go. You're live, Mel.
Okay. Yeah, Yeah, I've recently been reading something about there's some kind of connection between IgM and the COVID. And I've copied a couple of articles about it. It was actually in the scroll on CNN the other night, the COVID vaccine and IgM. I'm not quite sure I understand whether if you have IgM helps or hinders the effect of the vaccine. I was wondering if you had any information about this.
Yeah, good question. So again, remember, you've got, your body makes normal IgM, and it makes that in response to, remember when I spoke earlier about if you breathe in a virus, for example, your body's immune cells show it to B cells turn into plasma cells, and they make antibodies to that virus. The first antibody they make is an IgM. And then after a while, they make it a little bit more sophisticated, and they kind of, so again, you want to joke a little bit. The first thing they make is a dirty Scud missile. It just will go after anything that's vaguely looks like the same virus. But then after a while, they're going to be a little more refined, and then it's going to be a laser missile. It's going to go right to exactly what it's supposed to hit. So when they actually are checking to see if you've been exposed to coronavirus, what they're going to be doing is they're going to check for IgM first, because that's what you make first. And that's the quick way to know whether you've actually been recently exposed to COVID infection. On the other hand, if you were exposed a little while ago, you've now changed it over to that IgG, which is much more now specific. So important thing to know is IgM comes first. That's why they'll check that, because that allows them to know if your infection was recent.
So does that help the vaccine, or?
Nope. So all it does is it purely just tells us whether the person has some natural immunity so far. And we don't even know exactly what that means. In other words, is it enough immunity to stop you getting infected again? It just tells us you are exposed and are reacting or have reacted to the virus that you were exposed to. And it will tell you if you're reacting to the vaccine.
Thank you.
I have a question.
Joel Rosenblatt.
Yeah, I have a COVID-related question, or vaccine-related question. Before chemotherapy, they tell you not to take things that makes your cells stronger. You know, stop your vitamins, stop any other supplements you may be taking that make your cells stronger. Does any of that apply to a vaccine for COVID? Should you take your regular medications and supplements, or are there things you should stop taking?
So to the best of our knowledge, there isn't anything you should stop taking. And my view is you keep taking the things that you're taking, especially your medications and the like, because you don't want to suddenly find you have a heart attack because you stopped your medications to prevent that. You know, that would be unbelievably silly. The reason sometimes you get this discussion about stopping things that stimulate your immune system, especially at the beginning, is we don't remember Waldenstrom cells are part of the immune system. So when you are non-specifically or just generally stimulating your immune system, we don't know exactly what you're stimulating. So hopefully you're stimulating the good part of the immune system that fights the cancer, but you could just as easily be stimulating the cancer cells directly too. So sometimes that's where just being a little wise about some of these really immune stimulatory kind of things is important. However, when it comes to a threat like COVID-19, you know, we think that that threat is really dramatic, and anything we can do to make sure your immune system is in good shape, including the vaccine, that's the reason to go for it for sure.
Pat, did you have a follow-up question about IgM levels? Pat, New Mexico?
Yeah, so Pat, I see your message in the chat. What about if you don't make a lot of IgM? So if your IgM levels are very low. So many of you will know if you got a lot of treatment, if you got a lot of rituximab, it can make your immunoglobulins be low all round. And to be frank, that's a risk, because that says that your ability to mount the immune response you need can be compromised. And what's a challenge, many of you might get IVIG. So, that's basically replacing what you don't have. And it's replaced from healthy donors. The challenge, however, right now is we don't know how many of those healthy donors actually are making immunoglobulin against COVID. So when you get IVIG, we don't actually know how good that is in protecting you against COVID-19. So we don't think that you're probably going to have a totally normal amount of immune protection if you have a low IgM, but it's going to be better than nothing. So that's why we would still get the vaccine.
Doctor, I'm curious if the policy that you spoke about coming out of Mayo Clinic, is that something that they are only sharing with their own patients? Or is that something that could be used as like an advisory update for WM patients worldwide? If we, was that something that these support group leaders could maybe share in writing with folks? Because you were so unequivocal in saying, if you can get it, get it. That's not like, that's a phenomenal message to get out to everyone. I feel like there's an invaluable clarity. Do you?
So I can only say I don't speak for Mayo Clinic, and this came out to the Mayo Clinic Cancer Center. It wasn't a policy statement that went out to the broader community, but I don't think you're going to get too much pushback from other groups. I don't think you're going to hear a lot of different tones. To the best of my knowledge, there are virtually no groups around the country that are saying don't get the vaccine, except for folks that don't believe in the vaccine. But if you're looking at folks who see that as of benefit, folks, overall, everyone is being recommended, if you are able to get it, to get it. Now, again, have to be clear, there's no one that can require you to get it. So even at Mayo, where, for example, if I don't get my tuberculosis test, they will discontinue my employment. They have not said that for the COVID-19 vaccine because it's not actually fully approved. It's emergency use. But I wouldn't be shocked in a year or so. If I don't get an influenza shot, I can be in a lot of trouble because, again, risk to the population. But when it comes to COVID-19, it's voluntary at this point. But we strongly would recommend considering it.
It's very helpful to hear you say the as soon as possible piece, because although there might not be question as to whether to get the vaccine or not, a lot of us, maybe we've been trained in watch and wait, but we're just like, let's hold back, let the other kids go first. We'll see what happens to them. And then we'll get ours because we just want as much information as possible going into this and not put ourselves at risk. So just hearing you say, go for it is very clarifying and super helpful. I don't know if there's anybody else who wants to give a raise. Eileen Sullivan in Boston. I thought that those of us who've had low levels of monoclonal IgM still have normal levels of normal IgM. Yes or no?
And that is true. Yep. So for example, if you have, I'm making this number up, 200 milligrams per deciliter of IgM, it might be the actual amount of M protein or monoclonal protein may be very low, and almost all of that is normal. But there are some of you and others that you will know that will actually say, my IgM level is 5, and so it's like in the basement. Well, even if all 5 of them are normal, you're stretched pretty thin when it comes to natural immunity. And that does put you at some risk.
Interesting.
I would also say one other thing. So, I haven't had both my doses yet. I've only had one. But, I've had lots of other vaccines in my time. I certainly got yellow fever, and that just about paralyzed your arm for like a week. So this one was a breeze compared to that. It get a little stiffness, but certainly, as I say, no other real symptoms. I understand you get some more reactions with the second one, so we'll see.
Yeah, I was going to say, let us know after next week. We want to hear after the second one. We have one question about the steroid use prior to the vaccine.
Yeah, so, steroid use obviously can compromise your immune response. So again, if you're on chemotherapy, if you're on steroids, if you're on rituximab, you can't expect 95% efficacy of the vaccine. That's entirely true. However, we don't think it's 0. So I don't know what the number is in between, but the reason we recommend getting it is because it's better than nothing.
Excellent. I think there's a question about shingles in the chat, but I believe you just answered that. Shingles.
Yeah,
and-
so, Don, I believe, is saying, what's the question about shingles and COVID? We recommend a 2-week interval between the two.
Yeah, my question though, related to, I have the shingles vaccine at the beginning of January. I'm scheduled to get the second one the beginning of March. Can I get the COVID vaccine between the two?
Yes. And I would also say my personal view would be I would prioritize the COVID vaccine, and I would try and do that exactly 3 weeks or 4 weeks apart if you're either getting the Pfizer or the Moderna version, because there's a lot more we've done a lot more shingles vaccines, and we are pretty comfortable that if you go a few weeks this way or that way, with your second dose, probably doesn't matter. So, I think you could delay it if you needed to on the shingles side.
Thank you.
We've got another question about coming from Canada. What about ibrutinib and the vaccine? And then I see we have Judy in Texas wanting to ask a follow-up after that.
Yeah, so, ibrutinib obviously depresses the ability of B cells to make immunoglobulin. And so, yep, it's like rituximab, it's like chemotherapy, it's like steroids. All of those are going to have an effect on your ability to mount the immune response. So completely would agree that your immune response is not going to be the same as the next person who has no medications and is a healthy volunteer. But as I said earlier, you're not going to have zero response. It's going to be somewhere in the number above that. So we just think it's worth it for whatever that number is. And you heard me not say a number because I don't know what the number is.
That's great. Judy in Texas.
Dr. Ansell, thank you so much. I'm going to try to word this so that I don't put you on the spot and try to play.
I've never been worried about being put on the spot before.
But between the Moderna and the Pfizer virus, I've been reading a lot about the temperatures, and the chemical composition of these two. Is there a preference on how they work with your immune system? Is one better than the other? Is one more effective, or is one more dangerous? That's from a reaction standpoint.
Yep. So Coke versus Pepsi.
Yes.
Okay. So depends what you like. Depends. Everyone will swear out the one versus the other, but as a class, they work in the same way. They have a very similar benefit ratio. They have a very similar likelihood of creating immunity. So all told, I think the best one to get is the one you can get first. So it doesn't really matter to me which vaccine you can get. The one that you should take is the one you're being offered. And so.
And then you can go out to the CDC and get the drugs that are in. Can you get a list so that if there's anything you're allergic to? For instance, I got the flu vaccine and got really sick, and I found out they preserved it with thimerosal, which I'm allergic to.
Yep. So you could search. So again, they'll ask you if you've had those kind of reactions before, right before they actually give it to you. And if you have, they may sort of ask you more details. And if it turns out, the preservative or the suspending protein that they have it in is the one they give you the other one. So that's maybe true, but if the people that actually react to this based on an allergic reaction is probably 1 out of 100,000. It's a very low number.
Thank you.
Okay. We have a couple of interesting questions here. And thank you so much. This feels like shooting gallery, like we're just getting them coming left and right. Do you need a break? If you were in, I'd get you like a cup of coffee or something.
I got to walk a long way for my cup of coffee. So, hey, I'm watching 2:30. I'm going to head out of there and go get one.
I just want to be sure. I'm like, geez, this is really cruel. We're just putting you on the hot seat and we don't even give you some water. So should we be more cautious and get the COVID vaccine in a healthcare facility versus getting it in a Walgreens or CVS with everyone else?
Quite frankly, I think the biggest risk is you. So, the people that have been most likely to get ill are actually the people that don't wash their hands and the people that don't stay away from other people and the people who don't wear a mask. So yes, those other people are a risk to you, but if you're a savvy person, you're not standing right next to the guy who's coughing on you. You know, you're moving away in a hurry. So whether you get that in the healthcare facility or you get it at Walgreens, doesn't matter. You're still you're going to do the social distancing thing at the same rate each place. So I think, again, it's all about which place can you get to most efficiently. If you've got to drive 50 miles to go to the healthcare facility, whereas you could get at Walgreens in a mile, I think go to Walgreens.
Right. Okay. So that's super helpful. Let's do one last question because we want to be sure we get to the rest of the pre-submitted questions that are not COVID related, just because there were some really interesting ones there that I know folks are excited to hear your input on. The last one was related to the steroids. What you said several questions back was if there's any increased risk when you receive a steroid spinal injection. This is a very specific, this might be too specific for this purposes, but I just wanted to.
No, so again, it's a good question. So for example, if you get any medication that is in a specific place and it is a formulation, and usually it is, that limits it to that place, the risk in the rest of your body is pretty low. So what do I mean by that? If you rub a steroid cream on your skin, most of it is not absorbed, you know, because the goal is it's supposed to do good on your skin. It's not supposed to immediately get absorbed and, you know, make you fat and chubby because of too much steroids because it went into your body. So, you know, it's made to stay there. Similarly, if you're getting a steroid injection or a spinal, you know, a steroid injection, we use products that will not diffuse out of that area very easily. We want it to stay there because it's got to do good where you gave it. So actually, it has almost very little. You can never say none. In medicine, the word none and never, it never happens. But anyway, bottom line is it very seldom will actually go out of that particular location to somewhere else. And so it has a very limited effect on the rest of you.
Super helpful. Thank you so much, Doctor. Would you like to see in the other, the more general questions that we sent you in advance that were non-COVID related, if you want to jump in? I know there was a question about, well, there was a very specific question about ASH, and I'm going to pull them up in front of me.
Yep, so I'll just read them off and then. So there was a presentation at ASH that spoke about FGF, which is fibroblast growth factor effects on Waldenstrom's. And that's actually some interesting science. And you might remember when I spoke about the microenvironment and the place in the bone marrow environment where the cell lives. That's a cytokine, a protein that promotes the growth of the tumor cells. And that just creates another opportunity to target that as a way to potentially make the environment more healthy for good cells and less favorable for cancer cells. I think that's got a ways to go. There is an early molecule or two that may target that, but it's something to watch. And as I mentioned earlier, one of the exciting things in Waldenstrom's are all the new agents that are coming along. And my future I see for Waldenstrom's is combining these to get a better effect overall. There was another question about is there any research planned into understanding bone and soft tissue involvement by Waldenstrom's? I will say that's a little bit of a tricky question. And the reason it's tricky is when you have Waldenstrom's that's moved out of the bone marrow and out of the lymph nodes and out of the spleen, and it's in the soft tissue and it's in the bone per se, that needs a little harder look, because that's not how Waldenstrom's usually behaves. So one of the things we worry about is sometimes that's changed its nature to something like a large cell lymphoma. We call that a transformation. Or it's maybe not exactly Waldenstrom's, one of the very close cousins, things like marginal zone lymphoma. So that's where MYD88 mutation can help you, and a few other things. So all I would say, yes, there is research in that space, but it's not specific to Waldenstrom's because that's an unusual Waldenstrom's picture. Could I explain covalent drugs? So is there, can non-covalent BTK inhibitors be used as primary treatment? So again, those of you who can remember, ever did chemistry or other kinds of college level classes will remember covalent and non-covalent bonds. What does that mean? The one is it sticks pretty much permanently. And the other is it sticks on and off, on and off, on and off. So when you have a covalent bond, like a BTK inhibitor, what that's doing is it comes along to this protein that we're trying to mess with, and it basically goes in, there's a little pocket, it goes into the pocket and sticks there, and it never comes out. And that particular, only way you get rid of that bond is for that BTK to be broken down and go away. A non-covalent bond is, it goes in, but a little bit later, if it kind of gets a little disinterested, it may disengage, and then a little later on, it goes back in again. So it may bob in and out. Turns out, probably doesn't matter. The fact that it's just gone in there, messed with that function long enough that it stopped doing its job. So a lot of that covalent and non-covalent story and all the rest of it, I think, is more marketing and more theoretical than practical. For us, it's most BTK inhibitors are, again, the Coke and Pepsi analogy. They, everyone who's a Coke, aficionado would say there's definitely better, and the Pepsi guys would say that's definitely better. And quite frankly, it's somewhat theoretical. Next thing was, why isn't obinutuzumab used more often? So rituximab and obinutuzumab are very similar. Obinutuzumab is also a CD20 antibody that got engineered to be a little bit more likely to make the immune system work with it and go after and attack the cell it's stuck to. And I think the reason that it hasn't been used quite as much is there have been many studies that have been done comparing them head to head. And only a minority of them have shown that it's better. In low-grade lymphoma, there's a study specifically that shows that it may be better, especially when combined with chemotherapy. The problem, unfortunately, is it has a little bit more in the way of infusion reactions. And because some of the studies were negative, it became a little bit difficult to tell whether it really was an improvement. And a lot of people were very comfortable with rituximab, and so kind of just stuck with that. Also, if some of you might have received rituximab under your skin, or rather than as an infusion, which is actually really convenient because that's quick. It's like 15 minutes instead of a few hours. So that's another reason you can't do that with obinutuzumab. So that may be another reason it hasn't really got quite as much traction.
Oh, interesting.
There's a question here about getting bendamustine plus rituximab. Good response, 6 years. Would you be inclined to use the same treatment? The answer is it depends a little bit on how well you tolerated the bendamustine rituximab. It also depends on how many rounds of it you got, and it depends on how long it had a good effect. But if you got 6, 7, 8, 10 years from it, I'd certainly do that again. My hope would be you get that again. On the other hand, if you only got 2 or 3 years, I would consider a different treatment because, as I mentioned before, we got lots of very effective therapy, and some of the other medications may then get you more mileage than the bendamustine. And then last question that I see here is ibrutinib dosage. Do small people do better with lower doses? And that's a good question, and the answer is nobody's tested that. Basically, the way they worked out the dose of ibrutinib was they looked at, remember how I said there's that little pocket and the drug kind of gets stuck in there? They basically just went up and up and up and up on the dose until everybody on the clinical trial had 100% of that pocket filled with the ibrutinib. And they said it's called receptor occupancy. Every single BTK, every single receptor was engaged by BTK, by the BTK inhibitor. And then they said, that's your dose. But remember, I said sometimes they can pop in and out and it doesn't seem to matter. Well, many of you will know we go down on the dose and the results are still good. So do you really need all that drug? Don't know. So when all is said and done, I think, probably when you're smaller, probably many patients on it could use a lower dose. I think most physicians are just too concerned that they may compromise the outcome of the patient by doing that, and that's why we don't. I mean, quite honestly, a lot of research, and particularly drug sort of development, is done somewhat just empirically. I mean, I smile to myself, the dose of rituximab, it was not worked out any scientific way. They had 20 patients going on the study. They were giving them all 4 doses. They said, how much do we have? And they split it. the total amount by the number of people and the number of doses. And that's how we got to a rituximab dose. No science in that one.
Wow. Oh, wow. Is that true?
Dr. Trian and I were there. So he'll tell
Whenever
you.
you have a chance.
We were both there.
Wow. Never heard that one before.
Yep.
Somebody was going to say something, and then there was a question about getting MYD88 testing before.
Yeah, I was. This is Jim Bushy.
Hi, Jim.
I got one quick question. I was taking a larger dose of ibrutinib. My nephrologist found that it was attacking my kidneys. So I'm in actually kidney failure right now. And we reduced the dose to 140. And guess what? It works.
Now, I would say that's quite a good comment. You're a little bit special. You knew that already, sir, but now you know it for a fact. But you got to remember, when your kidneys are malfunctioning, you filter your blood slower.
Right.
So drugs circulate longer, which means the dose you may require is lower. So you're quite right. You getting 140 is working great, and that's fabulous. But you could also actually argue that effectively your kidneys, the way they're working, your dose is higher than that, because the other people would actually pee that out. You don't. It circulates longer, so effectively it brings your dose up. But your point you make is accurate. There are people with normal renal function or normal kidney function where we decrease the dose down to 140 because of irregular heartbeats and other things, and it still works. So you're right on.
Well, the reason why I asked is because I didn't have the renal problem until after I started taking the ibrutinib. Okay. Yep. And from what I've read, that there is a possibility, not a lot, that people do become, you can get renal failure from the drug. So I guess the question is this: why don't we start off at a lower dose and see how it performs before hitting everybody with such a high dose?
Yeah, so again, a good question. So two ways you could think about it. One is, if you come in low and it doesn't work enough and try and crank up, cells often become resistant. And so it's actually quite difficult. So often we come in high, get control, and then dial down. So obviously, if we did the damage already, then that's a problem. But quite frankly,
then let me ask you a question. Hypothetically speaking, because this happened to me, I was at 400 in my IgM. After 2 weeks of taking the drug, I had dropped below 1,000. 2 weeks, okay? So specifically at that time, wouldn't a protocol, are they going to start a protocol where you would start weaning you off the drug to see if it holds?
So we know for a fact that if you were to stop at that point or drop the dose down, but particularly if you stopped, it would bounce right back up within a few days.
Okay.
So we haven't done enough. The only time we start to get more comfortable about stopping the drug is based on data from CLL, chronic lymphocytic leukemia, where they have considered stopping the drug at around 2 years. But notice 2 years is quite different to 2 weeks.
Well, yeah, that's it. Exactly. Now, because I've been taking the drug for a lot more than 2 years.
Right.
Okay. So, Todd, so the 140, by the way, I didn't get a flare. I've been on the 140 dose now for probably 4 months.
Right.
And my IgM's holding at 900. It did go up a little, but not a lot. And I think I can live at 900.
Totally. No,
Okay.
That's the right choice.
All right, thanks,
I'm
Doc.
jealous of that 900, Jim. Can you send some of that over to Philly so I can use that? That's pretty good. You got me jealous now. Dr. Ansell, I think that wrapped up every single question that we not only submitted in advance, but we threw into the chat box and then just raised our hands to ask you. Is there anything else before we close that you wanted to add, or anything that you wanted to ask a group of 50 WMers in the room that you've always wanted to know but were afraid to ask?
Oh, I've never been afraid to ask anything, but thank you though. I will say this much: thank you to each of you for what you do, because quite frankly, information matters, and when you get out there accurate information about what WM and about what you should do related to COVID and the like, you really make the lives of healthcare workers easier. So thank you for what you do. Thank you for being a support group leader. Thank you for the support you provide people, because each of you know that these are life-changing diagnoses, and having a team around you that cares for you and that's interested in you and engaged with you matters. So again, thank you for what you do.
Dr. Ansell, thank you so much for the gift of your generous time, your warm presence, and this informative update today. You know, it's such a stressful and anxious time for folks, and to be able to spend this much time with you explaining it all to us in ways that we could understand is a gift beyond measure. I can't really express my fullest gratitude. You're really truly a treasure and a master teacher, and all WMers are so grateful. If you look through these two screens here, you'll see 50 people, but understanding that behind them are standing every WMer in their support group locally on the ground across the US and Canada. So they are the mouthpiece who will then take this incredible message that you've just given us and communicate it to those folks, and have enormous impact and ripples that just keep on giving. So thank you with a full heart, Dr. Ansell, for everything that you're doing, and please go on and continue doing it on our behalf. We're wishing you strength, and we don't know how you squeeze so much in 24 hours in a day, but however you do it, please keep doing it. And we just know you have our fullest gratitude. Let's unmute everybody.
Thanks,
so we can just applaud and say goodbye to Dr. Ansell appropriately.
Thanks very much, everyone.
Thank you,
Thank
Dr.
you.
Ansell.
Thank you.
Thank you.
Thank you.
Thank you. Thank you.
Thank you, Dr. Ansell.
All right, bye guys. Thank you very much.
Thank you so
Yeah.
much.
Thank you, Shelley. Bye-bye.
Thank you, Lisa.
How lucky are we, right? Do you know what I'm saying?
When will it be available?
To pick his
Yeah.
It will be available.